Hello, my name's Ben. I'm a Year 2 Biosci student from Cardiff Uni, and I'm just going to give you a little rundown of the Golgi Body and its role in the secretory pathway. So what I've done is I've made a lecture just outlining what the Golgi is, how it's built, what its functions are, and I hope you enjoy. So let's begin. So to understand how the Golgi Body processes proteins, its structure must be analysed first. The Golgi body is found near the nucleus of the cell, whilst also being in close association with the ER. So here I've drawn the nucleus, and there's the ER just attached closely to it, and then the Golgi body is in close proximity with the ER and nucleus. So the Golgi is made of flattened cisternal membranes, which resemble disc-like shapes. So here are those flattened membranes, and they're similar to discs. I guess they're a bit curved, but just refer to them as disc-like shapes. So when referring to a cisternal membrane, what I mean is it's a flattened vesicle which houses enzymes inside of its lumen. So one of these discs is called a cisterna, and inside a cisterna there are enzymes. These enzymes are found in the lumen, and the lumen is basically inside of the cisterna. So these enzymes are what modify the protein, so they're really key to the Golgi's function. And the whole structure is a Golgi ribbon. So the Golgi's flattened cisternal membranes organize themselves into stacks, which are connected. So these are the connections shown in yellow. This makes a continuous membrane system known as the Golgi ribbon. And the Golgi ribbon, even though you can't see it, it's held in place by microtubules. So as the Golgi is a stack of flattened membranes, that means it has two ends. So one here, close to the ER and nucleus, and one here, far away. So these two ends are really important as they have different characteristics and names, which are essential in the protein processing function of the Golgi. So the cisterna, found closest to the nucleus and the ER, is known as the cis face, or cismocisterna. In this region, one can see budding and fusing of vesicles. So vesicles would go from here to the cis, from the ER. So importantly, the cismocisterna is heavily fenestrated, meaning it has lots of small pores. These pores are present so that materials arriving from the ER can easily be deposited into the lumen of the Golgi body cisternae. As the cis face's main job is to receive materials from the ER, it is known as the receiving end of the Golgi apparatus, as it receives vesicles from the ER. So moving on to the opposite end of the Golgi, we find the trans face and the trans Golgi network here. So here, modified proteins are sent off to different parts of the cell inside coated vesicles. So at the trans side, one can see highly fenestrated tubules which extend from the transverse cisternae. So here's the transverse cisternae, the one that's furthest away, and here are those highly fenestrated tubules. So the transverse cisternae is the last and most furthest cisternae away from the nucleus and ER. The highly fenestrated tubules allow materials to easily leave the trans Golgi network. So now I'm going to zoom in on the Golgi and its relationship with the ER. So how do proteins go from the ER to the Golgi? So the ER and the Golgi have an intimate relationship. ER navigates the Golgi cisternae and is closely wrapped around the swollen regions of the organelle. This close relationship is demonstrated by the two organelles having membrane contact sites. So these membrane contact sites are where the fenestrations, or pores if you like, of the two organelles align. This alignment makes it easier for materials to pass through. And also importantly, these membrane contact sites are very, very small. They're like between 10 to 30 nanometers in width. So this short distance means there's not a lot of distance for the proteins to travel. So diffusion and other modes of transport is easy from the ER to the Golgi. So knowing the structure of the Golgi allows us to have a better idea of what it does and how it does it. So the main function of the Golgi body is to process and modify proteins arriving from the ER and then shipping these modified proteins off in vesicles to different parts of the cell. Some proteins go to other organelles, some go to lysosomes, and some go to the cell membrane ready for exocytosis. So I've mentioned that proteins are modified in the Golgi. What do I mean by modified? Well, proteins arrive at the cis face of the Golgi and undergo several possible changes catalyzed by the Golgi enzymes. These enzymes are responsible for molecular changes to the proteins, the most noteworthy being glycosylation, which is the addition of a carbohydrate, and then you've got phosphorylation, which is the addition of a phosphate group, and then you have sulfation, which is the addition of a sulfate group, and finally you have other proteolytic modifications, which basically means a breakdown of proteins. So these changes can change the original protein structure drastically. This can cause it to fold or even have different yet important characteristics for its function. Through these enzyme-controlled modifications, the Golgi can produce glycosaminoglycans, phospholipids, enzymes, sphingolipids, lysosomes, and many more molecules. Golgi's body's function can be simplified to like a sorting hub of proteins. So it receives proteins, it modifies proteins, and it repackages them to send them off to their destination. So the Golgi's enzymes are the one who carry out the modification processes inside of the Golgi. So the Golgi's enzymes are found in subcompartments, with their location depending on what is being processed. These compartmentalized enzymes are anchored onto the membrane surface of the cisternae. These anchored enzymes are known as resident enzymes. The advantages of this compartmentalization is that it allows optimization of pH, ion composition, and substrate concentrations in each compartment. The enzyme's location depends on what is being processed. So, for example, when glycoproteins are being processed, the enzymes that are needed are distributed in each cisternal subcompartment according to their mode of action. So what is meant by this? Basically, enzymes which remove a certain sugar, I'm going to think of one such as mannose, are found in the medial cisternae. So these are the mannose digesting enzymes. So these, if a protein would go through, these lot would remove or add mannose. I imagine remove. So whilst these enzymes remove mannose, other enzymes which remove other sugar chains could be found closer to the transcisternae. So how would a protein get from here to there? So we need to know, how do proteins move through the Golgi body? So we know that the Golgi is made of stacks of discs called cisternae, which house resident enzymes that modify proteins arriving from the ER and ships them off to other parts of the cell. But how does the protein enter the cis side, move through the medial, and exit the trans phase? So there are several models which try to explain how proteins are trafficked through the Golgi apparatus. The two most common proposed models are the vesicular transport model and the cisternal transport model. However, we first must look at how proteins leave and join the Golgi body. So proteins leave the ER at specific exit stations known as ER exit sites, or ERES for short. The ERES are morphologically different from the rest of the ER. The structure of ERES have been studied using a yeast species known as Saccharomyces cerevisiae. In this species, researchers found that the ER exit sites appear as numerous scattered points throughout the ER. More ER exit sites are observed and accumulated on the high curvature regions of the ER membrane. So what I've done is I've drawn the ER and see where it folds and curves. This is where the ERES, the ER exit sites, would accumulate. So this accumulation suggests that ER exit sites prefer to organise at the ER high curvature domains. The reason as to why ER exit sites are found in the high curvature regions of the ER may be due to the structure of certain proteins which make vesicles at the exit sites. So don't get too worried about these, but proteins such as SAR1 and Sec23-24 pre-budding complexes. So these proteins are what make vesicles from the ER to go to the Golgi, and these proteins are concave in shape. So basically they're saddle shaped. So these saddle shapes associate better with the curved membrane regions of the ER. So that's why I've drawn them like this, just to imitate the vesicle forming on the high curvature regions. So I talked about vesicles. If a protein wants to leave the ER and go to the Golgi, how are they packaged into vesicles? How can they go from the ER to the Golgi in a vesicle? So, proteins are packaged into vesicles known as COP2 vesicles. So the word COP2 refers to a type of vesicle coat that transport proteins from the ER to the Golgi apparatus specifically. Yeah? So COP2 travels in an anterograde direction. Anterograde is basically forwards. So how do they form? How do COP2 vesicles form? A receptor within the ER membrane binds to the protein that is going to the Golgi. This binding changes the conformation of the protein. So here the cargo protein is binding to a receptor on the ER membrane. So this conformational change attracts SAR1 GTPase, which is an enzyme. SAR1 is important as it encourages other things needed to make the vesicle to come together. So here's SAR1, here's the ER membrane, here's the cargo protein with the receptor. So SAR1 is attracted due to the changes in conformation caused by the binding of the cargo protein to the receptor. Sec12, a transmembrane protein, comes along and exchanges GDP for GTP. So here's the SAR1 and the receptor and the cargo protein complex, and here's Sec12, which comes along and exchanges GDP for GTP. After this, another transmembrane protein called Sec23-24 comes along and binds the ER membrane receptor with SAR1. So this binding attracts Sec23-24. So as you can see, it's sort of like a mushroom shape. It's starting to build that vesicle coat. And next, so after this, when the vesicle coat's starting to form, Sec13-31 comes along and multimerizes the complex. This means that it replicates all the bound proteins to form a vesicle. A curved coat is made as the vesicle forms. So here's Sec13-31. It replicates this structure, starting to form the outer coat of that COP2 vesicle with the cargo protected. So we know how the vesicles are formed. So how are the COP2 vesicles directed to the Golgi? How do they fuse when they arrive? The movement of the COP2 vesicles is directed by Rab proteins. Rab proteins are small guanosine triphosphates which regulate protein transport. They anchor themselves on the C-terminus and escort proteins and carry the vesicle through the cytoplasm. So there's the Rab anchored onto the vesicle. So The fusion of COP2 vesicles to the target membrane, which in this case is the Golgi membrane, is facilitated by snare proteins. Snare proteins are universal fusion proteins that regulate docking of vesicles to target membranes. So, here's the vesicle with the outer coat gone, and the snare proteins have basically caught the vesicle at the Golgi site, and this is where the protein cargo is released from the receptor and goes into the Golgi. So now, we know what the two vesicles do. We know what COP2 does, and we know how it leaves and how it gets there. Going back to the original question of how do proteins move through the Golgi, current evidence points towards a model known as the cisternal maturation model. This is where the protein stays in the same cisterna throughout the Golgi network, and enzymes are carried between each cisterna in a retrograde direction. The cisternal maturation model has three distinct stages, and we're going to start with one. So number one is the cis side assembly stage. So this is where the cis-most cisterna is made, how it's made. So let's have a look at it. So COP2 vesicles bud off from the ER and fuse homotypically to form the ER intermediate complex, or ERGIC for short. So not the ER, I meant the endoplasmic reticulum Golgi intermediate complex, ERGIC for short, but I'm going to call it the intermediate complex just to save some time. So this fusing happens multiple times. Okay, so COP2 vesicle fuse to form the ERGIC. And see, so the proteins from inside the COP2 vesicle, they fuse to form the ERGIC, and the ERGIC contains the protein cargo. So many COP2 vesicles fuse, making multiple intermediate complexes. So here are more COP2 vesicles, they also fuse, making more intermediate complexes with the protein cargo inside, shown by red. So these ERGICs again fuse and assemble to make the cis cisterna of the Golgi body. And the cis cisterna contains the protein cargo from the original COP2 vesicles. Sometimes proteins from the ER membrane get shipped off from COP2 vesicles by mistake, and they can end up in the cis cisterna of the Golgi. So to get rid of them, proteins that are not supposed to be there are shipped back to the ER by COP1A vesicles. These vesicles travel in a retrograde direction, meaning they go backwards. So I've mentioned COP2 vesicles, and now I've mentioned COP1A vesicles. So what are COP1A vesicles? So COP1A vesicles bud exclusively from the cis cisterna and occupy the space between the cis cisterna and ER export sites. This means that they move in a retrograde direction and carry ER resident enzymes back to the ER. So this is why I was talking about the COP1A removing those the enzymes that are meant to be in ER back from the cis to the ER. COP1B vesicles, another type of vesicle, and they bud exclusively from the medial and trans-Golgi cisternae and confine to the space around these cisternae. COP1B vesicles are really important as they carry Golgi resident enzymes from the trans and medial cisternae to younger cisternae in a retrograde fashion. So, if you think about it, there are enzymes here with the protein cargo inside, but these COP1B vesicles carry enzymes, not proteins, enzymes to each cisterna back retrograde direction. It's the same principle as the COP1A going backwards, but instead of going straight to the ER, they are found in the medial, trans and cis. COP1A is just found at the cis going back. COP1B goes backwards throughout the Golgi. So now we can go back to the cisternal maturation model after looking at these vesicles, and we now know what these vesicles can do. We know that COP1A goes backwards, we know that COP1B goes backwards. We can now go back and look at how the Golgi moves proteins through itself. So after formation, the cis cisterna transitions and loses the ability to receive COP2 vesicles, which are carrying the protein cargo from the ER. The cis cisterna gains the ability to receive glycosylation enzymes and sugar nucleotides carried in COP1B vesicles. Receiving these enzymes converts the cis cisterna into the medial cisterna. And from there, the medial goes into the trans cisterna. So this process repeats. Medial becomes the trans due to receiving late-acting enzymes from old trans cisterna. So as I mentioned before, the COP1B vesicles, they carry enzymes. So enzymes from the medial cisterna, they're meant to be there, they're living in the medial, they get brought back to the enzymes in the cis cisterna. And this makes the cis cisterna mature into the medial cisterna. So basically the cis becomes the medial becomes the trans, as enzymes are passed backwards throughout each cisternal region. Importantly, the medial and trans cisternae are involved in carbohydrate synthesis. So this is where enzymes modify the proteins by adding and or removing sugar chains. Due to the enzymes being compartmentalized inside the cisternae, early acting carbohydrate making enzymes reside in early medial cisterna. Late acting carbohydrate making enzymes reside in the late trans cisternae. And cisternae at this stage exchange enzymes again through COP1B vesicles. So I'm just going to explain this diagram that I've drawn. So here we have the cis-most cisterna. This is just basically recently formed from those endoplasmic reticulum Golgi intermediate complexes fusing together with all the protein cargo. And this is the later models. So in the early medial cisterna, it contains early medial acting enzymes. So these enzymes get passed back to the cis, and then the cis containing those enzymes becomes the early medial cisterna. And this process repeats, as mentioned before. The late medial cisterna passes on its late medial enzymes to the early medial cisterna. This causes the early medial cisterna to become the late medial cisterna, and then the trans cisterna passes on its late acting enzymes to the late medial cisterna, causing the late medial to become the trans. So it's this process of the discs of the cisternal membranes with these enzymes maturing as the enzymes are exchanged backwards. COP1B vesicles carry these enzymes to each cisterna backwards, causing each younger cisterna to mature. This maturation is where the cisternal maturation model gets its name from. The cisterna mature into older cisterna containing different enzymes able to act on different carbohydrate sugar chains. So after protein modification, we've got to get rid of them, we've got to send them off into the cell. So after modifying the proteins, transcisterna transitions so that it can no longer receive or produce COP1B vesicles. The transcisternae instead gain the ability to produce clathrin-coated vesicles. Clathrin-coated vesicles are selectively sought cargo to the cell membrane, trans-Golgi network and endosomal compartments for multiple membrane traffic pathways. The modified proteins bud off the trans-Golgi network and clathrin-coated vesicles and travel to their destination. So this is the transcisterna. As it said, it's matured so that it can no longer produce or receive COP1b vesicles. This means the enzymes inside of here can no longer go backwards, and enzymes from the older cisternae, so the transcisternae after this, they won't be accepted backwards. So the enzymes in here are localized and they cannot change. So this maturation changes what the transcisterna can do, and then the transcisterna gains the ability to produce clathrin-coated vesicles. These clathrin-coated vesicles are what carry the modified proteins all around the cell. So why is this model, why does it work? What do we have evidence for it? Well, I'm just going to tell you about it now. So, is there proof, basically, of the cisternal maturation model? And I can tell you there is. So, by analysing Golgi proteins, researchers observed changes in the enzyme composition of the individual cisternae over time. This meant that enzymes in each cisterna changed due to them being carried to younger cisternae by COP1b vesicles retrogradally. Another piece of evidence is that procollagen, a large cargo protein, traverses the Golgi stack without leaving the lumen of the cisternae. This contradicts the vesicular transport model, which uses vesicles to carry the modified proteins from each cisterna to the next in an anterograde direction. Remember, anterograde is forwards. That's what the vesicular transport model does. It carries proteins forward in vesicles from each cisterna to the next. That means enzymes in each cisterna are the same. So I'm just going to carry on talking about the vesicular transport model. I'm just going to give you a little brief overview of it again, because it's important. So what is the vesicular transport model? So this model refers to the use of vesicles to transport the proteins from one cisterna to the next, from cis to trans. For this medial to be sustainable, the Golgi apparatus must be regarded as a constant structure. So that means it doesn't have a maturation sort of model. It can't change itself. It's not fluid. So these enzymes inside the cisternae are localized, and the proteins are the ones that move, not the enzymes. So these proteins move through each layer of cisternae until they are sufficiently modified. Vesicles containing the proteins move in an anterograde direction, meaning they move forward up the Golgi towards the trans Golgi network. So they go from cis to trans in vesicles to each cisterna, and they get modified by the different enzymes in each cisterna. So after looking at the vesicular transport model, there must be other proposed Golgi transport models. I'm just going to give you two of them. So there is the cisternal maturation model with heterotypic tubular transport. So this model follows the same principles as the cisternal maturation model, but has some additional features. The main feature is that there are tubular connections between the stacked cisternae, forming the Golgi ribbon. These tubes allow bidirectional flow of proteins and enzymes between the Golgi cisternae. This allows fast traffic of materials. The drawback of this model involves the transport of a large protein like collagen. So due to collagen size, these small tubular connections are not suitable for the transport of a large cargo protein like collagen. It just wouldn't work. So I'm just going to explain this diagram. So here we have a protein inside the cis side, and here are these bidirectional flowing tubes, and these allow the materials to go both ways. So this would mean that proteins are carried fast throughout this Golgi network. But this model isn't sustainable due to tubular connections not supporting the transport of a large cargo protein like collagen. So another proposed Golgi transport model that's quite new is the rapid portioning in a mixed Golgi. Rapid partitioning. So this model says that Golgi has two separate domains in which the processing and export of proteins occurs. Cargo proteins from the ER move between these two Golgi domains, and they are able to randomly exit at any level of the Golgi to travel to their destination. So this model allows large and small proteins to exit the Golgi bodies. These are the advantages. So this would allow collagen to move in and out. However, it has no mention of how the two domains form, or like how are they maintained as separate things. Why aren't they just together? How can they just be separate? And how would the enzymes be exchanged? There's no mention of this. Between the cisternae, so are these enzymes exchanged here? Are the enzymes exchanged between domains? So these two domains form one Golgi body, but they're still separate. There's still a space between them. So we've got the drawbacks, we've got the advantages, and now I'm just going to look at some new research in the field of the Golgi. So by now you should have a better understanding of the Golgi, what it does, how it does it. But there is an interesting new approach as to how COP2 vesicles are incorporated into the Golgi body. So it's the hug and kiss action. Mentioned in this lecture is that COP2 vesicles accumulate and fuse to form the ERGIC, or the ER-Golgi intermediate complex. However, there may be another way in which COP2 vesicles are incorporated. The hug and kiss action describes how the cis-Golgi network picks up the COP2 vesicle and its contents from the ER exit sites. The cis-Golgi envelopes the COP2 vesicle, which is bulging from the ERES with its cargo attached. The coat of the COP2 vesicle is removed, exposing the cargo proteins. The cargo proteins then fuse with the cis-Golgi membrane. After this, the cis-Golgi detaches with the cargo protein. So I'm going to run you through this diagram again because it's quite confusing. So here is the COP2 vesicle, and here is the cis-cistern. It comes along, it envelopes that vesicle, removing these coats, the COP2 coat from the vesicle, just leaving the receptor binded with the cargo protein. And then for simplification, I imagine it doesn't pick up the receptor. I imagine it just picks up the cargo protein, but to make it seem quite understandable, I've drawn it like this. So the cis-Golgi fuses with the protein cargo and just takes it away. So it hugs, there's the hugging motion. Kissing is that exchanging and taking it away. So that's the hug and kiss model. So I hope you've enjoyed that lecture. It might have gone really fast for you, but I hope it was alright. Here are some references for each section if you want to have a further read. I recommend you do. They probably have a lot more detailed explanations of what I've tried to tell you. So we've got overview, Golgi, functions, Golgi body enzymes, proteins, and there's more there. So thank you for listening. I hope you've enjoyed, and yeah, thank you.